Part 3, SPMs, the Biology of Resolution. Chapter 7, What are Specialized Pro-Resolving Mediators, SPMs? Specialized pro-resolving mediators are the lipid-derived signals the body uses to actively complete inflammation without simply shutting immunity off. That definition has to be stated carefully because SPMs are often misunderstood from the start. They are not the same thing as general anti-inflammatory compounds. They are not blunt suppressors. They do not function by making the immune system weak, silent, or indifferent. They function by changing the phase of the inflammatory response. They help move tissue out of recruitment, escalation, permeability, and collateral damage, and into clearance, containment, repair, and restored control. In practical terms, SPMs are part of the body's exit intelligence. They are how the organism stops behaving like it is still in the middle of an emergency when the useful work of that emergency has already been done. The phrase specialized matters because these mediators are not generic background lipids. They are produced through defined enzymatic pathways from specific polyunsaturated fatty acid precursors, especially EPA, DHA, and in some cases N3-DPA. They belong to distinct mediator families such as resolvins, protectins, maresins, and lipoxins, each of which contributes to different parts of the transition from inflammatory force to inflammatory completion. The phrase pro-resolving matters because they do not merely reduce signal intensity. They direct work. They reduce further neutrophil recruitment when further neutrophil recruitment is no longer useful. They enhance efferocytosis, the macrophage-driven clearance of apoptotic cells and inflammatory debris. They help restore barrier integrity, limit unnecessary tissue damage, reduce persistent pain sensitization, and support the return of tissue from emergency metabolism toward normal function. The phrase mediator matters because these are real signaling molecules, not vague wellness concepts. They carry instruction through receptor-mediated biological effects, and those instructions change what cells do next. This is what separates SPMs from the older and much more limited view of inflammation as a process that simply rises and then fades. For a long time, inflammation was described as though it naturally burned out once the provoking stimulus weakened or once enough anti-inflammatory pressure was applied. That picture left out one of the most important discoveries in modern inflammatory biology, which is that resolution is not passive. It is an organized biochemical phase with its own signals, its own timing, its own receptor systems, and its own cellular tasks. Tissue does not automatically return to baseline just because the early inflammatory burst slows down. Dead cells still have to be removed. Extracellular debris still has to be cleared. Vascular permeability still has to normalize. Nociceptive amplification still has to be withdrawn. Immune traffic still has to stop arriving in excess. Barriers still have to be rebuilt. SPMs are part of what makes that happen. Their importance becomes obvious once chronic inflammation is viewed correctly. Many chronically inflamed tissues are not failing because they lack activation chemistry. They are often rich in activation chemistry. They can recruit, amplify, leak, sensitize, and escalate with impressive consistency. The deeper problem is that they do not transition well. They remain suspended in inflammatory mid-process. Neutrophils continue arriving or continue leaving behind unresolved debris. Macrophages fail to clear efficiently or fail to shift fully into restorative phenotypes. Cytokine tone remains elevated. Oxidative chemistry remains active. Membranes remain unstable and continue releasing more substrate for early-phase signaling. The tissue behaves as though the emergency is still open because in biochemical terms, the signals required to close that emergency have not gained enough control. SPMs matter because they are part of the signal system that closes the loop. This also explains why SPMs should never be framed as simple immune suppressors. Suppression reduces force. Resolution redirects force. That distinction is central to this entire book. A suppressed immune system may produce less overt inflammatory output, but that does not mean tissue has been cleared, danger has been removed, or repair has begun correctly. In some situations, suppression can even delay completion by muting signals without restoring the cleanup phase that must follow them. SPMs do something more precise. They reduce the continuation of unnecessary recruitment while increasing the efficiency of clearance. They restrain collateral damage without preventing the body from defending itself. They help terminate pain-promoting chemistry while encouraging removal of the dead cells and debris that would otherwise keep generating that chemistry. They do not erase inflammation as a concept. They help finish it as a sequence. Because they are lipid-derived mediators, SPMs also reinforce one of the major themes of the book. The body stores part of its future inflammatory and reparative language inside membranes. EPA and DHA are not valuable only because they improve membrane composition in a static sense. They are valuable because once incorporated into phospholipids, they provide precursor material from which pro-resolving mediators can later be generated. This means the capacity to resolve is partly built ahead of time. A tissue carrying more useful omega-3 derived substrate is carrying more of the raw material required for later exit signals. That does not guarantee successful resolution on its own because downstream conversion, oxidative stability, immune phenotype shifts, and sufficient metabolic support are all still required. But it does mean the ability to produce SPMs is rooted in prior structural conditions, not just in the heat of the moment. SPMs also clarify why the common phrase anti-inflammatory is too blunt to describe what many tissues actually need. A tissue trapped in chronic inflammatory activation does not only need less inflammatory signaling, it needs better inflammatory sequencing. It needs the later instructions that tell neutrophils to stop arriving in excess, tell macrophages to increase efferocytosis, tell the vascular bed to stop behaving like an emergency corridor, and tell nociceptive systems that the phase of heightened protection can now be withdrawn. That is the territory of SPMs. They belong to the completion phase, not merely the silencing phase. They represent one of the clearest examples in biology of how the body solves problems not just by turning processes on and off, but by moving them through phases in the correct order. The different SPM families also matter because resolution is not one task, it is many tasks coordinated together. Resolvins help reduce ongoing inflammatory cell recruitment and influence multiple parts of tissue de-escalation. Protectins are associated with tissue protection, especially in neural and other specialized contexts, and with limiting inflammatory injury. Maresins are strongly linked to macrophage-mediated repair, efferocytosis, and restorative tissue behavior. Lipoxins, though often derived through different precursor relationships than the omega-3 derived families, are also part of the pro-resolving landscape and demonstrate that the body has multiple routes for signaling that the acute inflammatory phase must not remain dominant. The point is not that each family should be memorized as a separate trivia category. The point is that the body has built an entire class of mediators whose function is to end inflammatory work intelligently rather than merely to oppose inflammation in the abstract. This chapter matters because it marks the transition from talking about omega-3 as substrate to talking about what that substrate can become. EPA and DHA are not the finished answer. They are upstream materials. SPMs are part of the downstream answer because they are among the mediators generated from those materials that actively help the body regain order after disruption. They reduce unnecessary continuation. They improve cleanup. They protect tissue from prolonged inflammatory injury. They help restore the structural and signaling conditions that ordinary function depends on. In a body that can still initiate strongly but resolve poorly, SPMs help explain what is missing. The organism does not only need less force, it needs the biochemical intelligence required to know when force has done enough. Resolvins. Resolvins are one of the clearest examples of why inflammation cannot be understood only through the language of suppression. Their role is not simply to make inflammatory signals smaller. Their role is to help the body change phases. They are lipid mediators generated from omega-3 fatty acid precursors, mainly EPA and DHA, and they belong to a class of signals produced after those fatty acids have already been incorporated into membranes, released under the right conditions, and converted through downstream enzymatic pathways. This makes resolvins part of the body's active exit strategy. They do not erase the need for early inflammation. They begin to matter when early inflammation has already done enough, and the tissue now needs to stop recruiting damage faster than it can clear it. In that sense, resolvins are not peacekeeping molecules in the sentimental sense. They are operational molecules that reduce unnecessary continuation and improve the conditions required for completion. The name itself is instructive. Resolvins are called resolvins because they participate in resolution, not because they merely lower inflammatory intensity in a vague way. They help reduce further neutrophil infiltration once the useful phase of neutrophil-heavy recruitment is ending. They influence macrophage behavior in ways that improve debris clearance and support the removal of apoptotic cells before those cells break down and become new inflammatory fuel. They help limit collateral tissue injury caused by prolonged inflammatory traffic. They can reduce pain-promoting signaling and help shift vascular and tissue behavior away from open emergency mode. This is what makes them biologically important. A tissue trapped in chronic inflammatory repetition often does not need weaker immunity. It needs better instructions for when to stop escalating and start clearing. Resolvins belong to that instruction set. There are two major families typically emphasized in this context: E-series resolvins, which are derived from EPA, and D-series resolvins, which are derived from DHA. That distinction matters because EPA and DHA do not simply duplicate one another. Each supplies a different precursor pathway, and therefore contributes to a somewhat different mediator landscape once conversion begins. E-series resolvins represent one route by which EPA-based incorporation can later become pro-resolving signaling. D-series resolvins represent one route by which DHA-based incorporation can later become pro-resolving signaling. The practical point is not that one family must be treated as superior in the abstract. The practical point is that omega-3 in tissue becomes more biologically meaningful when it is understood as stored precursor for families like these, rather than as a static nutrient badge. The membrane holds the possibility. The resolvin is one form that possibility can take once the body is ready to move from force toward restoration. Resolvins also show why timing is everything in inflammatory biology. A molecule that reduces neutrophil recruitment too early could impair useful defense. A molecule that reduces neutrophil recruitment after adequate early phase work has been done can prevent unnecessary tissue damage and help the system move into cleanup. Resolution is therefore not the opposite of immunity. Is immunity shifting tasks? Resolvins do not tell the body to ignore disruption. They tell the body that further escalation is no longer the smartest use of energy and tissue. That distinction is central. In chronic inflammatory states, the problem is often not that the body cannot defend itself. It is that the body cannot stop defending as though the emergency were still expanding. Resolvins begin correcting that error by changing what kind of work inflammatory cells are being asked to do. This is why resolvins matter so much in the broader logic of omega-3 biology. EPA and DHA are not endpoints. They are upstream structural and signaling precursors. Resolvins are one of the downstream mediator families that show what omega-3 is for. It is for building a body that can still initiate when necessary, but does not remain trapped in initiation when the relevant phase has passed. A membrane reservoir containing omega-3 derived substrate carries more than a nutritional fact. It carries future access to mediators like resolvins that can help the body reduce further inflammatory recruitment, improve clearance, protect local tissue, and regain order. Without that downstream possibility, omega-3 remains only partly expressed. With it, the tissue gains one of the main biochemical tools required to finish what inflammation began. Protectins. Protectins are DHA-derived specialized pro-resolving mediators that help defend tissue during and after inflammatory stress without relying on blunt suppression. Their name reflects that role. They are called protectins because they participate in tissue protection, especially when inflammatory activity, oxidative stress, or cellular injury threatens to convert a necessary response into unnecessary damage. This makes them especially important in a book like this because they show clearly that resolution is not just about making inflammation smaller. It is about preserving tissue while the inflammatory sequence is being brought under control. A response can begin appropriately and still become destructive if the tissue undergoing that response is not protected from prolonged exposure to its own defense chemistry. Protectins belong to the part of the body's mediator system that helps prevent that slide. Like other SPMs, protectins do not appear from nowhere. They depend on prior membrane incorporation of DHA, later release of DHA from phospholipids under the right conditions, and downstream enzymatic conversion into a mediator form the tissue can actually use. That makes protectins a good example of why DHA should never be treated as a finished endpoint. DHA is valuable structurally, especially in neural and retinal tissue, but its value also lies in what it can become. When tissue is under stress, protectin generation gives the body access to a class of signals that can reduce unnecessary inflammatory injury, influence leukocyte behavior, and support the shift away from open-ended inflammatory escalation. In that sense, protectins are not just downstream products. They are part of the reason storing DHA in membranes matters in the first place. Their protective role is especially important in tissues where inflammatory collateral damage is costly. Neural tissue is the clearest example. The nervous system can tolerate remarkably little disorder before function begins to degrade. Excess immune recruitment, persistent oxidative chemistry, excitotoxic stress, prolonged membrane disruption, and unresolved debris can all damage signaling precision and structural continuity in ways that do not stay local for long. Protectins are important here because they help create conditions under which tissue can survive inflammation without remaining trapped in inflammatory injury. They belong to the logic of controlled defense rather than runaway defense. The body still responds, still detects danger, still performs cleanup, but it does so with more regard for preserving the integrity of the tissue that has to remain functional afterward. This is also why protectins should not be understood as simply weaker inflammatory mediators. They are not watered-down versions of initiation signals. They represent a different phase and a different set of priorities. Early inflammatory mediators help recruit, amplify, and alter tissue conditions quickly. Protectins help reduce the continuation of that damage when continuation is no longer useful. They support the turn from disruption toward containment and preservation. In practical terms, they help the body limit how much of the tissue has to be sacrificed in order to solve the problem that triggered inflammation in the first place. That is a very different job from merely lowering inflammatory volume. It is a job of strategic restraint in service of repair. Protectins also reinforce one of the most important themes running through this manuscript, which is that membrane composition determines future behavior. A tissue enriched in DHA is not only building a certain kind of membrane, it is building a certain kind of future response potential. If inflammatory stress occurs, the tissue has more access to DHA-derived downstream mediator families, such as protectins. If that substrate is sparse, or if oxidative damage keeps destabilizing the membrane environment in which it is stored, the tissue may remain relatively poorer in the signals that help protect it during the later phases of inflammation. This is one reason chronic inflammatory environments are so destructive. They do not only activate too much initiation chemistry, they often fail to generate enough preservation chemistry to prevent the inflammatory process from injuring the tissue it is supposed to protect. Seen clearly, protectins help explain why DHA matters beyond the familiar language of brain support or membrane fluidity. DHA is not valuable only because it sits inside neural tissue. It is valuable because under the right conditions it can be converted into mediator classes that help defend that tissue from the consequences of unresolved inflammatory stress. Protectins are part of the body's way of saying the response must now become more selective, less damaging, and more compatible with long-term tissue survival. They do not eliminate immune activity. They help refine it so that defense does not continue shredding the environment it was meant to preserve. Maresins. Maresins are DHA-derived specialized pro-resolving mediators produced primarily through macrophage-centered pathways, and they are especially important because they connect resolution directly to cleanup and repair. Their name comes from macrophage mediator in resolving inflammation, which is exactly what makes them so useful conceptually. They show that resolution is not merely the decline of inflammatory intensity. It is organized work carried out by cells that have shifted roles. Early in inflammation, immune activity is dominated by recruitment, containment, and force. Later, if the sequence is functioning properly, macrophages begin taking on a different kind of responsibility. They clear apoptotic cells, remove debris, help limit ongoing tissue damage, and support the transition into reconstruction. Maresins belong to that later phase. They are part of the signal language through which the tissue begins moving from combat into restoration. This macrophage connection matters because macrophages sit at the hinge point between unresolved inflammation and genuine repair. A tissue can recruit neutrophils rapidly, generate eicosanoids efficiently, and still fail to heal if the macrophage phase never becomes effective enough to clear what the early phase left behind. Dead cells that are not removed become new inflammatory stimulus. Damaged membrane fragments, oxidized lipids, extracellular debris, and unresolved microdamage all keep telling the tissue that the emergency is still active. Maresins help interrupt that cycle by supporting the kind of macrophage behavior that closes inflammatory work rather than perpetuating it. They improve efferocytosis, support debris handling, and help reorient the tissue away from continued escalation. This makes them central to the argument of the book because chronic inflammation often persists not only from too much initiation, but from too little successful cleanup. Like resolvins and protectins, maresins depend on prior DHA availability. They are downstream products, not spontaneous events. DHA must first be incorporated into membranes, stored within phospholipid architecture, and then released and converted under the right conditions. That means maresin biology begins long before a macrophage starts clearing anything. It begins with tissue composition. A membrane environment that contains usable DHA is carrying part of the precursor pool needed for later restorative signaling. This is why omega-3 should never be understood as a finished answer at the moment of intake. Its value lies partly in what it can later become once the body enters the stage of inflammation, where cleanup and repair must take priority. Maresins are one expression of that later value. They are what DHA can become when the tissue is ready not merely to stop inflammatory recruitment, but to start resolving the structural consequences of what recruitment has already done. What makes maresins distinct is their strong association with tissue restoration rather than with de-escalation alone. Resolvins are often discussed in relation to limiting further inflammatory recruitment. Protectins are often emphasized for their role in preserving tissue from inflammatory injury. Maresins, while overlapping with both, point even more clearly toward rebuilding conditions. They belong to the biology of repair-compatible resolution. A tissue cannot truly resolve if it only stops bringing in new inflammatory cells. It also has to remove what has accumulated, restore local order, and reduce the debris burden that would otherwise keep provoking the next round of inflammatory activity. Maresins help create that transition. They make inflammation more finishable. This is especially important in chronic disease states where tissue seems to remain stuck in a loop of irritation, partial damage, incomplete recovery, and rapid reactivation. In many of those settings, the body is still capable of signaling danger, but is less capable of clearing the aftermath of danger. Macrophages remain present, but their behavior may stay skewed toward inflammatory participation rather than restorative clearance. The tissue remains chemically noisy because the remnants of previous inflammatory cycles have not been removed well enough to let the next phase begin cleanly. Maresins matter here because they are part of the downstream signal environment that helps turn macrophages into better finishers. They improve the quality of the handoff between defense and reconstruction. Seen clearly, maresins reinforce one of the deepest ideas in this book: resolution is labor, not silence. The body does not return to health by merely reducing inflammatory output. It returns by clearing the remains of what inflammation did, restoring structural order, and reducing the likelihood that leftover damage will become the trigger for another round of activation. Maresins are part of the biochemical language that supports that work. They are DHA-derived instructions for a tissue trying to regain coherence after stress. They help explain why omega-3 matters not simply for calming the body, but for helping the body clean up after itself well enough that it no longer has to keep acting inflamed. Derived from EPA and DHA. Specialized pro-resolving mediators are derived from EPA and DHA because these long-chain omega-3 fatty acids serve as the stored precursor pool from which the body builds part of its resolution chemistry. That point is more important than it first appears. It means the body does not improvise resolution from nothing after inflammation has already caused enough disruption. It has to enter the inflammatory sequence already carrying the structural material required for a controlled exit. EPA and DHA must first be absorbed, transported, incorporated into membrane phospholipids, and retained in tissue long enough to become part of the cell's future signaling reserve. Only then can they be released under the right conditions and converted into mediator families that help reduce continued neutrophil recruitment, improve macrophage-driven clearance, protect tissue from excess collateral damage, and restore local order. Resolution, therefore, depends on prior membrane composition. The tissue can only derive these signals if it was built with the right precursor material before the need for those signals became urgent. EPA and DHA do not generate the same downstream products, and that difference matters because it shows that omega-3 biology is not one undifferentiated category. EPA is the precursor for E-series resolvins, which participate in reducing ongoing inflammatory recruitment and shifting the character of inflammatory signaling away from simple continuation. DHA is the precursor for D-series resolvins, protectins, and maresins, which extend the resolution program into tissue protection, efferocytosis, and repair-compatible macrophage activity. This means EPA and DHA are related, but they are not functionally identical. Each supplies a different branch of the downstream mediator landscape, and those branches help solve different parts of the same biological problem. The body is not just trying to make inflammation weaker. It is trying to change what the tissue does next. that requires more than one kind of precursor input. This precursor relationship also explains why omega-3 should never be mistaken for an endpoint. EPA and DHA are upstream molecules. Their presence in tissue matters because of what they make possible later. First, they change membrane behavior. Then they change substrate availability. Then, under activation, they can be released and enzymatically converted into SPMs. Then, those SPMs can change immune cell behavior, vascular behavior, nociceptive signaling, clearance efficiency, and tissue recovery. The real biological effect is therefore layered across time. A person does not benefit from EPA and DHA simply because the molecules are present in circulation or because they appear on a lab panel. The body benefits when those molecules have been incorporated deeply enough, preserved well enough, and converted effectively enough to alter the trajectory of an inflammatory event once that event begins. This is one reason omega-3 results are often less immediate and less uniform than supplement language implies. If SPMs are derived from EPA and DHA, then several things must already be true for that derivation to matter. The tissue must contain the substrate. The membrane must be able to release it appropriately. The enzymatic environment must be able to convert it into useful mediator classes. The inflammatory sequence must reach a point where those mediators can actually shift the tissue away from continued escalation. A chronically stressed tissue can fail at any of these stages. It may incorporate too little DHA or EPA. It may remain dominated by arachidonic acid-heavy initiation signaling. It may suffer enough oxidative damage that highly unsaturated membrane lipids become fragile before they become useful. It may remain too repeatedly re-triggered for the later resolution phase to gain command. In those cases, the limitation is not the concept of omega-3 itself. The limitation is that precursor availability has not been successfully translated into downstream resolution behavior. Seen clearly, the phrase derived from EPA and DHA carries a much larger meaning than a simple biochemical source note. It means resolution is built from stored membrane material. It means the body's capacity to exit inflammation depends partly on what kinds of fatty acids have already been loaded into tissue. It means omega-3 matters because it provides the raw material for mediator families that help inflammation finish rather than simply intensify. And it means the quality of the body's recovery is partly determined long before recovery begins because the membrane already contains, or fails to contain, the precursor pool from which the exit chemistry must later be made. Not suppressive, actively resolving. One of the most important distinctions in this entire subject is that specialized pro-resolving mediators are not suppressive in the blunt pharmacologic sense. They do not solve inflammation by simply shutting the immune system down, muting all defensive signaling, or forcing tissue into artificial silence. Suppression reduces output. Resolution changes the phase of a response. That difference is not semantic. It is biological. A suppressed inflammatory system may produce fewer visible signals, but that does not mean damaged material has been cleared, dead cells have been removed, vascular permeability has normalized, pain sensitization has been withdrawn intelligently, or tissue architecture has begun rebuilding in an orderly way. A response can be quieter and still remain unfinished. Specialized pro-resolving mediators matter because they do something more precise. They help the body complete the inflammatory sequence rather than merely dull its expression. This is why the word actively matters. Resolution is work. Neutrophil recruitment has to stop when further recruitment is no longer useful. Macrophages have to increase efferocytosis so apoptotic cells are cleared before they break apart and become fresh inflammatory stimulus. Debris has to be removed from the extracellular environment. Barrier integrity has to be restored. Vascular behavior has to shift from emergency access back toward controlled selectivity. Pain-amplifying chemistry has to be withdrawn once its protective role is no longer justified. These are not passive events that happen automatically once inflammatory force declines. They require instruction. SPMs are part of that instruction. They do not merely make inflammation weaker. They help direct the biological labor that turns an inflamed tissue back into a functional one. This distinction is what separates resolution from anti-inflammatory suppression. Suppression is often useful for symptom control, but it does not necessarily tell the body how to finish what it started. It may reduce cytokine output, reduce pain, or reduce swelling, while leaving unresolved debris, barrier failure, immune missequencing, and tissue instability still in place underneath the quieter surface. An actively resolving tissue is different. is not just less inflamed, is moving toward completion. Cellular traffic changes. Cleanup improves. Collateral damage falls. The tissue is no longer organized around escalation, is organized around exit. That is why SPM biology is so important. It restores the idea that the goal is not simply to make inflammation smaller. The goal is to make it finish properly. Seen in this light, omega-3 derived resolution pathways are not valuable because they sedate the immune system. They are valuable because they help the immune system stop behaving as though the emergency is still expanding when the relevant phase has already passed. They preserve host defense while reducing unnecessary continuation. They support immune competence while improving inflammatory termination. They do not cancel the defensive sequence. They complete it. That is the deeper meaning of not suppressive, actively resolving. The body does not heal by being silenced. It heals by being guided out of emergency with enough precision to clear the damage, preserve the tissue, and return to order without remaining trapped in defense. The missing half of inflammation biology. For decades, inflammation biology was taught and discussed as though the main story ended with activation. A trigger appeared, inflammatory pathways turned on, immune cells were recruited, vascular tone changed, permeability increased, pain sensitization rose, and the task of medicine became identifying how to reduce or block those outputs. That framework captured the beginning of the sequence, but it left out the second half, which is why so much modern thinking about chronic disease remains incomplete. Inflammation does not become pathological simply because it exists. It becomes pathological when it cannot finish. The missing half of inflammation biology is the biology of resolution, the active, signal-dependent, substrate-dependent process by which the body stops further unnecessary recruitment, clears cellular debris, removes dead cells, restores barrier integrity, reduces collateral damage, and returns tissue to a state that can support normal function again. Without that half, the picture of inflammation is structurally broken from the start. This omission matters because it changes what chronic inflammation appears to be. If the only biology that matters is initiation, then persistent inflammatory disease looks like a problem of excess force. The body seems to be overreacting, overproducing inflammatory mediators, and overactivating immunity, so the obvious solution becomes suppression. But once the missing half is restored to the model, a different interpretation becomes possible, and often more accurate. Many tissues in chronic disease are not failing because they cannot generate activation. They are often very capable of activation. They fail because activation is not being handed off to completion. Neutrophils continue arriving or continue leaving behind material that has not been cleared. Macrophages do not fully transition into effective efferocytosis and restoration. Membranes remain damaged and keep supplying new initiation substrate. Barriers remain open and continue feeding fresh triggers into the system. The body is not simply too inflamed. It is caught in unfinished inflammation. This is why so many symptom-centered strategies fall short. If the missing half of inflammation biology is ignored, treatment naturally gravitates toward turning early-phase chemistry down. That can relieve pain, swelling, heat, or biochemical markers of inflammatory output, and sometimes that is necessary. But it still does not answer the deeper question of whether the tissue has completed the inflammatory job. Has the debris been cleared? Has the vascular bed normalized? Has the nociceptive amplification been withdrawn because danger has truly decreased, or because signaling was muted while the underlying problem remained? Has the barrier been repaired? Has the membrane reservoir shifted away from automatic reinitiation? Has the tissue regained the structural conditions needed to remain quiet without constant intervention? Without the resolution half of the model, these questions disappear, and with them disappears the difference between silence and completion. The discovery and elaboration of specialized pro-resolving mediators changed its landscape because it showed that resolution is not passive decay, is an active program. The body generates specific mediator families from stored lipid precursors, especially EPA and DHA, that help shift immune behavior, reduce ongoing recruitment, improve efferocytosis, protect tissue, and restore local order. This means inflammatory completion is not a fortunate accident. Is built into the same biological intelligence that built inflammatory initiation. The body has start signals and finish signals. It has escalation chemistry and closure chemistry. It has molecules that open emergency access and molecules that tell the emergency machinery when to stand down and clear the site. The missing half of inflammation biology is therefore not a side note. It is the exit architecture, without which the entry architecture becomes dangerous by default. This missing half also explains why omega-3 has been so persistently misunderstood. When inflammation is viewed only through the initiation lens, omega-3 is interpreted as anti-inflammatory, which makes it sound like a nutrient that somehow turns inflammatory volume down from the outside. But within the fuller model, omega-3 becomes something more important and more coherent. Is resolution substrate. It provides precursor material for mediator families that help the body complete what arachidonic acid-heavy early-phase signaling helped begin. That does not make omega-3 the opposite of inflammation. It makes omega-3 part of the later sequence of inflammation. The absence of that conceptual shift is one reason the public conversation has stayed shallow for so long. The real value of omega-3 cannot be understood until inflammation itself is understood as a two-phase problem: initiation and completion. Seen clearly, the missing half of inflammation biology is the reason chronic disease so often feels stuck. The body still knows how to enter defense. It no longer exits efficiently. It continues recruiting, leaking, sensitizing, oxidizing, and signaling as though the event were still active because the later biology required to terminate that state has not taken command. Once resolution is restored to the model, many modern pathologies start to make more sense. Chronic pain is not just excessive nociception. It can be persistent failure to withdraw protective amplification after useful danger has passed. Chronic vascular irritation is not just excessive inflammatory tone. It can be repeated failure to normalize permeability and endothelial behavior. Chronic immune activation is not just an overzealous system. It can be a system repeatedly beginning without successfully finishing. The missing half is what explains why the body can be both highly reactive and poorly healed at the same time. That is why this half of the story changes everything. It moves the question from how to silence inflammation to how to make inflammation finish properly. It reframes omega-3 from supplement mythology into substrate physiology. It shifts attention from simple blockade towards structural remodeling, mediator balance, barrier repair, membrane composition, mitochondrial support, and immune phenotype transitions. Most importantly, it restores the idea that the body is not designed only to defend. It is designed to defend and then return to order. When that second design is ignored, medicine sees only half the event. When it is restored, inflammation stops looking like a one-way march toward damage and begins to reveal its real architecture, a sequence that is only healthy when it has an exit.